Welcome to this topic on Wobble-Base hypothesis or Super-Wobble-Base pairing. Crick proposed the Wobble-Base hypothesis in 1966 to address the discrepancy in the number of genetic codes and tRNA molecules. In addition, the human mitochondrial genome revealed only 22 tRNAs to read the 61 sense codon, which led to the concept of Super-Wobble-Base pairing. This topic is relevant in understanding the reading of the messenger RNA by a limited number of tRNA molecules during translation. In this video, we will explore features of the wobble base, super wobble base, pairing, and hypothesis. To understand the topic, we should learn first wobble base with the help of this cartoon. Shown here are 64 genetic codes including 61 sense codons. In most cases there are 2 or more codons for the same amino acid. Such a group of codons are known as degenerate codes. The first two bases of degenerate codes are identical while the third base is variable. This variable base is known as wobble base because it can pair with the first base of the anticodon in a non-standard manner. Therefore, the third base of a degenerate code and the first base in the tRNA anticodon is wobble base. Next, we will review features of wobble base. Let us review the features of wobble base in the tRNA. A wobble base in tRNA is present in the anticodon arm or anticodon as the first base. When I say first base, it is the base toward 5' end. When I say third base, it is the 3' end base. So this could be inosine or modified base, or it could be normal base such as A, C, G, U. However, the most frequent one is inosine. Let us examine the features of wobble base in messenger RNA. Wobble base is present always as a third base in a group of degenerate codons. This is a group of 4 genetic code which are degenerate code because the third base is variable, the first two bases are same. The wobble base is shown by the arrow here. This is always normal base which could be A, U, C or G. Before we move forward to hypothesis, let us discuss normal base pairing versus wobble base pairing. According to normal base pairing rule, A binds with U, C binds with G and vice versa. Any other combination is a wobble base or non-standard base pairing. For example, in this case you have 4 genetic code here. They are read by tRNA. The first two tRNA are binding or reading perfectly according to the base pairing rule, A with U, C with G. However, in the last two cases, these last two tRNA, they are not following the rules. A is going with C, G is going with G. This is violation. This is wobble base pairing or non-standard base pairing. And this will help you to understand the now wobble base hypothesis. Let us discuss wobble-base hypothesis. I will present first basic overview, then I will come to specific points with examples. This was proposed by Crick in 1966 to explain the discrepancy in the number of genetic code and tRNA known at that time. According to this hypothesis, the third base of genetic code can pair in a non-standard manner, means it can violate the basic rule of base pairing. Therefore, the third base is known as flexible or wobble base. This flexibility allows a single tRNA to read 2 to 3 genetic codes. Now let me explain specific points of this hypothesis with examples of normal and wobble base pairing. Before I explain the first example, let me explain the concept of first base and third base very clear because I will be using these terms repeatedly. When I say first base, it means the base toward 5' end. When I say third base, this is the base toward 3' end. And this is because whenever you read DNA, RNA, you start reading from 5' to 3' direction. So now let us take the example. When you have C or A as the first base of the anticodon, it can bind with the third base as G or U of the corresponding genetic code. This is normal normal base pairing, there is no violation, and therefore in this situation one tRNA will read only one genetic code. However, if you have U as the first base in tRNA anticodon, this will bind with A or with G at the third position of the genetic code. UA is a normal base pairing, whereas UG is abnormal base pairing, vowel base pairing, and this flexibility allows a single tRNA to read two genetic codes. Similarly, if you have G as the first base of anticodon arm, it can bind with C or U at the third position of the genetic code. GC is normal base pair, and whereas GU is abnormal base pairing, and this flexibility allows a single tRNA to read two genetic codes. Let us see the last example where I is the first base of the anticodon arm. I is deaminated adenine modified base. This can bind with C, with A, with U as the third position base in the corresponding genetic code. So this allows the maximum flexibility, and a tRNA therefore can read three genetic codes. So this is all about Wobble-Base hypothesis. According to this, I can summarize that a single tRNA can read maximum 2 or 3 genetic codes. So far we have discussed Wobble-Base pairing, which is related to cytoplasmic system. Now we turn toward the Super-Wobble-Base pairing, which is related to mitochondrial system. In mitochondria there are 22 tRNA molecules or 22 tRNA anticodons. In this case, the first base of the anticodon can base pair with any of the four bases such as A, U, C, G, and this allows flexibility of binding or reading of four genetic code by a single tRNA molecule. This is known as super-wobble base pairing, and such tRNA is known as super-wobble tRNA. So in summary, Wobble base operates in cytoplasm, whereas Super-Wobble operates in mitochondrial system. Wobble allows reading of 2 to 3 genetic codes by a single tRNA molecule, whereas Super-Wobble allows reading of 4 genetic codes by a single tRNA molecule. And in the end, I would like to thank you very much for watching this video. Please like it, share it, and subscribe it. This is a higher level content, and therefore I would like you to review our video on genetic code. Until next time, you take care. Bye bye.